The following is a selected video from masterthecontent.com, where you will find an extensive video library of lectures for a variety of standardized admission tests. We offer over 600 hours of detailed video lectures for a multitude of standardized tests. Use our interactive in-lecture table of contents to find specific topics of interest. Work through numerous in-lecture examples to help you internalize concepts. To learn more, visit masterthecontent.com. Your career, our passion.
The first thing that we are going to be looking at is Keto-Enol Tautomerism. So there's a pretty succinct definition of Keto-Enol Tautomerism on the screen right there. And it says that ketones and other carbonyl compounds that have a hydrogen at the alpha position are in equilibrium with a constitutional isomer that is referred to as an enol. Okay, so that's a very brief and succinct definition of that process. Now, what I want to draw your attention to is the fact that carbonyl compounds in general will be proton donors, or they'll behave as weak acids in the presence of strong bases. So when you have a strong base around, carbonyl compounds will tend to be weak acids or behave as weak acids in that they will donate protons. And that's exactly what we have happening at the bottom of the screen right here. So we have a generic carbonyl compound right here that is a generic aldehyde. And what is happening is that we have a generic base removing or abstracting a proton from the alpha position. Now remember what the alpha position is. It's the position once removed from the carbonyl group. So this is a carbonyl group right here, and that would be the alpha position. That is the alpha carbon right here. So that's an alpha hydrogen, and removal of one of those will result or results in the formation of what is referred to as a resonance stabilized enolate, a resonance stabilized enolate. Now we did encounter this ion in part 2 of this series, so you should be familiar with it. And what I want to point out in this case is if we were to reprotonate this intermediate right here, we could do so at two main sites. Okay, so we could either do so at the carbon, which would effectively restore the original carbonyl compound, or we could do so at the oxygen, which actually gives us an entirely new or entirely different compound. So if we actually do protonate this compound at a different site, at the oxygen atom, we get what is referred to as an enol. And that's what we have on the screen right there. We have an enol. So an enol is basically an alkene on which one of the carbons is attached to, or to which one of the carbons is attached to a hydroxyl group. So you pretty much have an alkene that has a hydroxyl group that is attached to one of the carbons involved in forming the double bond. Now, I want to point out two things in this case, okay? Two things have happened in the formation of this enol. The first one being the location of the proton has been changed or transferred. Okay, so as you can see in our original carbonyl compound, we had a proton attached to that alpha carbon. We removed it and restored it or placed it on the oxygen atom right there. So we effectively transferred a proton from one part of the molecule, one part of the structure, to another. Now the second thing I want you guys to take note of is a simple fact that we also relocated the double bond. And this is very plain to see. As you can see in the original carbonyl compound, we had the double bond right there between the carbon and the oxygen. And in our enol and the tautomer, we have the double bond between those two carbon atoms right there. So we've relocated the proton, and we have also relocated the double bond. And that essentially is what tautomerism is. So the enol and the original carbonyl compound are pretty much related by the transfer or the movement of a proton from one part of the molecule to another, and the movement of the double bond from one part of the molecule to another. So that's pretty much what we mean by Keto-Enol Tautomerism. Now this reaction can be catalyzed either in a basic medium or in an acidic medium. So what we're going to do next is we're going to move on and look at the mechanism of this reaction in a basic medium.